Good morning, class. So today our topic, we have two topic to discuss today. We have DNA replication and PCR. Okay. So remember in our past lesson, we have the DNA and the DNA, of course, made up of two strands, and we have the priming. We have the 5 to 3, and yung partner niya is 3 going to 5. The 5 here stands for the position of the phosphate group. The 3 here stands for the position of the hydroxyl group. Okay. DNA replication is a semi-conservative process in all cell. Why? As you could see here, we have the parent DNA or yung old DNA, and each newly synthesized DNA, meron silang copy ng one parental strand at yung newly synthesized strand. Okay, that's why it's called semi-conservative. So, another thing to remember for DNA, the important enzyme for DNA synthesis, though marami sila, pero yung number one, of course, DNA polymerase. So take note, the word DNA, yun yung molecule, polymer, like long material, polymerase, ASE, that's the name of the enzyme. So DNA polymerase is yung siya yung gumagawa ng chain of DNA. And the action of polymerase, as you could see, pwede lang siyang mag-add ng new nucleotide if there is a hydroxyl part dito sa ribosugar. Okay. Otherwise, if there's no if there's no OH here, hindi makapag-add yung DNA polymerase ng nucleotide. And the material or ingredients for DNA synthesis, of course, we have the DNTPs, YNTPs. We have deoxy, and then yung nucleotide natin, and then triphosphate. So we have different DNTPs. We have DTTP, DGTP, DCTP, and DATP. Okay. And during the reaction, of course, we have cleavage nung ating 2-phosphate group or yung tinatawag nating pyrophosphate. And during that time, 2 energy-rich phosphate bonds are consumed when adding each nucleotide. Okay. So kung makikita nyo dito, we have 2 DNA strands, ang 5 going to 3. 5 going to 3. I don't know why the label here is 5. And then we have 3 going to 5. Okay. No, this is 3 going to 5, then 5 going to 3. Okay. So, take note, this portion of the DNA, or ay tinatawag natin siyang DNA replication fork. DNA replication fork, we have a lot of enzyme there, and this is also the site of the DNA replication. Okay, so what are the important enzymes needed during DNA replication? Of course, we need helicase. Another is primase. And then we have DNA polymerase number 3, or DNA polymerase. We also need some protein called DNA strand binding protein. Okay. So, discuss natin isa-isa kung anong mga enzymes na to. So, simplihan muna natin. So, helicase, you remember the DNA, like helical structure, we need to unzip the DNA. So ito yung parang zipper, siya yung nag-open ng DNA, nag-unzip. And then, during the unzipping event, itong na-unzip na strand, pwede siyang bumalik ulit. So para hindi bumalik yung hydrogen bond, this strand are actually stabilized by single-strand binding protein. Okay? And then, pag na-unzip na yung DNA, we could put primer, which is made up of RNA. So yung additional primer is made by made by primase. Okay. So when we have RNA primer, pag may RNA primer na tayo, itong yellow-orange na color, makakapaglagay na yung DNA polymerase ng nucleotide or glowing DNA nucleotides. Okay? So, dun muna tayo. So, discuss natin ano tong lagging strand and leading strand. So, again, we have helical DNA. The one that unzip the DNA, of course, we have the helicase. And then, kung mapapansin nyo dito, we have the topoisomerase or DNA carries natin. So, usually, pag pinubuksan nyo tong DNA or nagka-unzip, sometimes there's a torsion here. So, itong DNA carries, pwede siyang mag-stabilize ng DNA. Okay? So, balik ulit tayo dito sa RNA helicase. So, when we have RNA helicase, what happened is pwede na maglagay ng primer. Okay? The one that adds the primer is RNA. The one that adds RNA primer. Of course, we have the DNA primase or the primase. Okay? And then to stabilize this single strand DNA, we have, of course, a single strand DNA binding protein. Okay. So, pansinin nyo itong strand. So, yung old strand dito, ito yung 3 going to, wait. Ito yung 3 going to 5. That's the old strand, yung dark green. And then yung old strand dito, itong light green, yung 5 going to 3. Okay. So, paano sinisynthesize itong 3 going to 5? So, take note, this is 3 going to 5. Therefore, yung complementary strand na kailangan is 5 going to 3. Okay? So, 5 going to 3. So, yung RNA primer mag-a-add ng, DNA primase mag-a-add ng RNA primer. And then, once we have RNA primer, pwede nang maglagay or gumawa yung DNA polymerase number 3 ng bagong strand. Okay? So, why is it important? Bakit mahalaga yung RNA primer? So, at the end portion of the RNA primer, itong primer natin is made up of RNA nucleotide at yung carbon number 3 of the ribosugar of this RNA may OH group. So, kung mapapansin nyo, yung DNA polymerase, pwede lang siyang mag-add ng DNA nucleotide dito sa growing point or yung carbon number 3 na merong hydroxyl group. Okay? So, ang synthesis, of course, 5 going to 3. So yung 3 part, yung hydroxyl part, continuous yung masisynthesize. Like, lagay lang ng lagay yung DNA polymerase ng DNA nucleotide. Okay, that's why this strand is called leading strand. Okay, yung newly synthesized is leading strand. And then yung template needed to make the leading strand. Of course, we have the leading strand template. Yung leading strand template, that is yung 3 going to 5. And then yung leading strand natin is lagging 5 going to 3. Okay. So, yung isa naman, lagging strand. So, pag napapansin nyo, yung direction of the synthesis is from right to left. So, tandaan nyo na yung RNA, yung primer, yung DNA primase, maglalagay siya ng RNA primer, and that direction would be 5 going to 3. Kung napansin nyo dito, itong old strand natin or parental strand of the DNA is 5 going to 3. At yung strand needed or complementary strand for here is 3 going to 5. But take note, yung DNA polymerase could only add growing DNA nucleotide dun sa hydroxyl group na sa carbon number 3. Kaya dito tayo maglalagay ng primer somewhere sa gitna, not sa dulo. Kasi kung papansin nyo, ito yung dulo, yung OH dapat. So, paano kaya makakapaglagay ng growing strand ng tuloy-tuloy? Ito yung 5 to 3, 3 to 5, 3 to 5, at yung 5 ay phosphate group. Hindi kayo makapaglagay ng growing DNA kung yung last part is phosphate. So therefore, pabaligtad siya. That's why in direction of synthesis papuntang left side. Okay. So maglalagay tayo ng primer here. 5 going to 3, then DNA polymerase can add yung nucleotide sa OH part, okay, and then synthesize. So that's why this strand, we call it lagging strand, dahil mabagal yung paggawa ng template na to because we need more primer, okay. And then we have here tau. So yung tau subunit, it holds 2 DNA polymerase and here, okay. So pag may question class, tanong nyo lang or i-chat nyo ako or send nyo sa email ko yung question kung medyo hindi maliwanag yung topic.